Hi, everyone. Good morning, good afternoon, good evening. Thank you so much for joining. I'm still waiting to for some more folks to join. Let's give them a couple more minutes to kind of figure out their connections and things like that. We'll start in 2 more minutes, 2 to 3 minutes. If you'd like to go get some coffee, tea, please do so. Thank you. All right, I think we can get started. Thank you once again for joining our inaugural Silantes webinar series. We are so very excited and honored to have you here. And also we're honored to have our inaugural speaker, Dr. Junji Iwahara, with us today. A quick bio intro about Dr. Iwahara. Dr. Iwahara has graduated from University of Tokyo, working with Dr. Yokoyama, and he also worked in some of the influential folks in the NMR world, Dr. Robert Clubb in UCLA, and also Dr. G. Marius Clore from NIH. We are honored to have you, Dr. Iwahara. Please take it away. Thank you so much.
Thank you, Ravi. I really appreciate this opportunity for sharing our recent work with other people. Today, I'm going to talk about basically isotope labeling method for DNA using isothermal amplification. As you know, DNA is a genetic material in biology, but in terms of chemistry and biochemistry, DNA is much more useful than just genetic materials. DNA oligonucleotides can be used for diagnostics using PCR, or therapeutics using aptamers to target particular proteins, using in vitro selection and evolution, you can create DNA oligonucleotides that can specifically bind to particular proteins. And you can use the same kind of methodology to create catalytic DNAs called DNA enzymes. And you can also use DNA oligonucleotides for nanotechnologies and produce DNA nanostructures or nanomachines. And you can even use DNA for DNA computing. And in principle, heteronuclear multidimensional NMR spectroscopy should be useful for these areas of research using DNA. However, the use of such heteronuclear NMR for these materials is quite rare. Okay, here is some statistics about DNA isotope labeling. And you can see that DNA isotope labeling is strikingly rare compared to RNA isotope labeling. Yeah, here, this is the numbers of NMR structures in PDBs for DNA and RNA solved by NMR spectroscopy. And the total numbers are kind of comparable. But if you look at the numbers of structures with isotope labeling, it's strikingly different. DNA isotope labeling is very rare. You can see the same thing in the statistics for BMRB resonance assignments. Okay, so here, this is guanine iminoproton and thymine or uracil proton resonances. And if you look at proton resonances, they are kind of comparable. But if you look at the nitrogen 15 resonances deposited to BMRB, DNA resonances is much fewer, okay, strikingly fewer than RNA N15 resonances. So obviously, there is a problem about DNA isotope labeling. Of course, there are some other effects. DNA assignment can be easier than RNA assignment because of the presence of well-dispersed 2' 2" resonances and methyl resonances. But that doesn't really account for this kind of huge difference. And in fact, there is kind of obvious difficulty in isotope labeling for single-stranded DNA, particularly. Okay, for RNA, the methodology for labeling is well established, and people use in vitro transcription with T7 RNA polymerase together with carbon-13 N15 labeled RNTPs. This is a well-established method, and everything is commercially available. And the good thing about this methodology is that the transcription reaction turns over. Okay, so the polymerase use the same template again and again to produce many copies of RNA, right? And isolation of the product is also quite straightforward because the size of the template and RNAs are quite different. And also the RNA is not associated with DNA, so you don't have to really separate by denaturation. Compared to this quite nice method for RNA isotope labeling, DNA isotope labeling is much more challenging for single-stranded DNA. Okay, so this is the most common way to make double-labeled single-stranded DNA, and this is what we call single extension approach. Because in this approach, the enzyme conduct the extension reaction only once for each template. Okay, so in this approach, a hairpin DNA is typically used, and this is a special DNA with an RNA residue at the 3' terminus. And the enzyme extend the strand. Of course, if you use double-labeled DNTPs, this part of the DNA will be carbon-13 N15 labeled. And after extension, just a single extension for each template molecule, the reaction mixture is basically subjected to alkaline cleavage at high pH and high temperature. And a nick is introduced between the RNA 3' residue and the product. And after cleavage, chemical cleavage here, denaturing page is used to separate this product from the template. This step is extremely challenging, especially for relatively long DNA, because even in the presence of high concentration denaturants, they can still interact. And if you use something like sequence gel shown here, still the presence of complementary strand makes the mobility quite broad. The band becomes very broad due to the interaction. This approach in principle works, but in fact, in practice, it works usually only for relatively small DNAs, like less than 20 nucleotides. For some other nucleotides, DNA structures like G quadruplex, application of this one is challenging. Maybe it's not impossible, but it's really challenging. Another reason for the difficulty is that sometimes the template, single-strand portion of the template, can form secondary structure, and that can inhibit extension by the polymerase. That's another problem. So there are several problems about this approach. Basically, there is no reaction turnover, so you need to use a lot of template. And you have to use a special template containing RNA as a chimera, I mean DNA RNA chimera. And template secondary structure matters for this method. And denaturing page is quite painful, and usually the yield is not so great anyway. Basically, this method is not really simple. It is labor intensive, and some special skills are required. It's quite difficult. Okay, of course, some of you think that DNA has a very nice features like PCR, right? You can amplify DNA quite easily using PCR. And in fact, a PCR was used for DNA labeling, and it works nicely. However, this methodology works only for double-stranded DNA. If you would like to make double-labeled G quadruplex, for example, this method, PCR-based method, doesn't work. Simply because all the products are in the form of double-stranded DNA. So we really need a good, better method for carbon-13 N15 labeling of single-stranded DNA. Because many of important non-canonical DNA structures are for single-stranded DNA. For example, G quadruplex is one example. Another example is I motif. And there are many DNA aptamers that show quite unusual structures. And the DNA enzymes also adopt quite unusual structures, like shown here. And for these single-stranded DNAs, we definitely need a better methodology for isotope labeling. And single-strand DNA labeling is also useful even for double-strand DNA, because by using that, you can selectively only one of the two strands, and that will reduce the number of signals. Okay, we came up with a new methodology shown here. This is what we call PaNDA, palindrome-nicking-dependent amplification. In this method, we use 3 macromolecular components shown here. One of them is what we call input DNA. This DNA has basically 2 components. One is palindromic sequence that forms a homodimer or maybe hairpin. And the other part is template for the polymerase. And this input DNA forms this kind of duplex at the palindromic region. And the enzyme called strand-displacing DNA polymerase extend the strand using DNTPs. But this enzyme is special polymerase, and it can also displace existing strands bound to the template. The other macromolecular component of this reaction is nicking enzyme. This enzyme recognized a particular sequence and introduced a nick. In this design, the recognition site of the nicking enzyme is present in the palindromic region, and nicking enzyme introduce a nick at the junction between the original DNA and the product. Okay, once nick is introduced, this strand-displacing DNA polymerase can extend the DNA, displacing the prior product. So when this reaction occurs in a cycle in this way, basically single-strand DNA are accumulated, and basically you can get a lot of copies of single-strand DNA from this reaction. Good thing about this approach is that, okay, in this case, the template is used many times. It's not single extension approach. Reaction turns over, so you get a lot of copies from the same template. And this is an isothermal amplification. You can use just a single temperature, 55 degree actually, and isolation of the product is straightforward because, you know, in this case, the majority of the product is already in a free state. So you can just use non-denaturing conditions for anion exchange chromatography and isolate this product quite easily. So this is more like RNA isotope labeling. You know, as I already mentioned, for RNA isotope labeling, people use in vitro transcription system, and for that, reaction turns over, and product isolation is straightforward. The same thing is true here. This is basically an amplification method, and product isolation is quite straightforward, even for single-stranded DNA. Basically, it was possible due to special features of strand-displacing DNA polymerase and a sequence-specific nicking enzyme. Okay, so this is an example of isolation of single-strand DNA from PaNDA. And this is just non-denaturing anion exchange chromatography, and basically this is the peak of the product, and this is the peak of extended double-stranded DNA, and you can easily isolate the product. Here is the reaction conditions we typically use for PaNDA. The input DNA concentration is 1 micromolar, and these are the concentration of polymerase. And for DNTPs, we use 0.2 millimolar, which is actually quite low concentration compared to the concentration in PCR. But it was kind of important to use low concentration of DNTPs. Anyway, if you ask about that, I can show some data on that. Another point is that we use relatively high salt concentration here for basically suppression of 3' overhang attachment to the DNA products. Of course, if you do isotope labeling, you have to use isotope labeled DNTPs, and for that we purchase from Silantes. Okay, so here is some data about 3' overhang attachment. As I mentioned, basically, if you use high salt concentration, you can get basically the DNA strand as you expect without any additional residue at the 3' end. But if you use low salt concentration, like 50 millimolar KCl, you get some molecular species with additional residue at the 3' terminus. And that was obvious in NMR spectra as well. Okay, so this is basically produced by PCR method using restriction enzymes. And in this case, both strands are labeled. Okay, so this is basically single-stranded DNA labeling by PaNDA. And compared to this one, we saw additional peaks like this when we used low salt concentration from the for the PaNDA reaction. And these additional peaks are due to 3' overhang, and the population of such species was about 30%. It's not much, but still, in some experiments, this could be problematic. So to resolve this problem, we used high salt concentration, and at high salt concentration, the polymerase basically dissociates from the terminal position before 3' and overhang attachment happens. So in that way, we can largely suppress 3' overhang. So typically we use this condition. However, I should mention that for relatively long DNA, like over 50 nucleotides, the use of such high salt condition may produce a lot of byproducts of shorter DNA. So in such a case, you may have to use lower salt concentration at the risk of having 3' overhang. Okay, we applied PaNDA first to telomere G quadruplex. In this case, the G quadruplex forms basically in multiple G quadruplex conformations. In NaCl conditions, there is one major conformation, but for KCl conditions, multiple conformations can be formed, and that was obvious from just simple HSQC spectra. Okay, so this is N15 HSQC and carbon-13 HSQC, and for the NaCl conditions, we saw just expected numbers of HSQC signals for each case, but in KCl conditions, we saw about 3 times as many signals as you see here. So you can easily tell that there are multiple species under this condition. We also used PaNDA to produce thrombin aptamer NU172. And this is a therapeutic aptamer used in clinical trials at phase 2. And this is the crystal structure of NU172 bound to thrombin. We produced carbon-13 N15 labeled NU172 using PaNDA. This is basically reaction results. And we wanted to see whether or not NU172 can form this kind of structure, unusual structure, even in the free state. Okay, so this aptamer has various non-canonical structural motifs in addition to Watson-Crick base pairs. This part is G quadruplex, and there are 2 reverse Hoogstin base pairs, and there is also TTA triad motif stacking on top of the G quadruplex, and there is additional GG mismatch here, and proteins bound to basically this region. Okay, and G quadruplex in this case has 2 layers of G tetrad. And usually for G quadruplex, there are 3 or more layers. So we wondered if this kind of structure is possible only in the presence of protein. Okay, we did many NMR experiments, double resonance or triple resonance experiments to assign NMR signals from NE172, and we found that this aptamer can form G-quadruplex even in the free state. The reason why we can say that is that we saw basically N15 and proton chemical shift characteristic for G-quadruplex here. And when we assigned these signals, they were indeed from the region for which crystal structure shows G-quadruplex in the complex. So even in the free state, this aptamer can form a G-quadruplex. We also confirmed the presence of a reverse Hoogsteen base pair in NE172. We used HNN COSY for the experiment, and we observed the direct evidence of hydrogen bond scalar coupling between the N7 of adenine and imino nitrogen of thymine for this one and this one. So this represents the direct evidence of Hoogsteen base pairs, because usually N7 do not show this kind of couplings. Okay, we also analyzed the behavior of NE172 in human serum. This is quite relevant to the therapeutic application of NE172 because the target molecule of NE172 is thrombin, which is present in, of course, blood. We made salt-free lyophilized NE172 sample and then dissolved the powder of this aptamer in human serum, and we did some NMR experiments. The data quality was relatively poor due to molecular crowding environment, but still we were able to observe some signals characteristic for the G-quadruplex. And because of the nuclease activity in the serum, the signals gradually became weaker, like this. So we are able to see this kind of effect by NMR as well. So we confirmed the presence of G-quadruplex of this therapeutic aptamer in the serum. That's quite interesting. But we should also mention that G-quadruplex state is not 100% at physiological environment. Actually, when we measured carbon-13 HSQC, we saw multiple species, and probably the G-quadruplex state is about 50% at 37 degrees. And we even observed exchange cross peaks in these exchange experiments, and we got kinetic information. We haven't published it yet, but we will do it soon. Okay, and we also used a PaNDA for DNA enzyme, DZ5C. This enzyme, DNA enzyme, was reported by ETSCON group recently, and we confirmed the catalytic activity of DZ5C produced by PaNDA, and it is indeed catalytically active. And we measured NMR spectra for the free state and substrate bound state for this DNA enzyme. And here, this is an overlaid spectra for the free DNA enzyme and complex with the substrate RNA. As you can see, the spectra are very different, reflecting the structural change of the DNA enzyme upon binding to RNA substrate. Okay, probably some of you are wondering how much cost is really required for this labeling. right? Okay, here's an example. For NU-172, to get 2 milligrams of purified material ready for NMR experiments, it costs about $800. But the majority of the cost is not DNTPs, labeled DNTPs, but enzymes. Okay, basically, enzyme costs more compared to the double labeled DNTPs. But still, this is basically 5 times cheaper than the commercial service of full labeled DNA. So this also makes economic sense as well. All right, so here is some conclusions. This method, which we call PaNDA, allows for easy enzymatic production of single-stranded DNA, and PaNDA also greatly facilitate isotope labeling. Therefore, it promotes NMR research on non-canonical DNA. Yeah, this work has been already published, and everything I showed today is already available in the paper indicated here. So if you know, you would like to know more about the work, please check this paper or contact me. Here is my research team. C1 did the most of work for the PaNDA reaction optimization, and Being Han did NMR work, and Shuhei also did NMR work. And in his case, he assigned signals of NU-172, and he also produced carbon-13 anything labeled NU-172. And Jonathan basically checked the reproducibility of the reaction. And I would like to thank again Ravi for this opportunity, and also for useful information regarding the cost for isotope labeling. And this work was funded by NIH and the Welch Foundation. And finally, I thank you for your attention.
Fantastic. Thank you, Dr. Iwahara. It's so good to see the research in depth. I know we talked about the price and all that, the NTPs and things like that. I remember, it's amazing that the new technique is almost 5 times cheaper, and even NTPs are not the majority of the cost. It's good to see we are not the ones who are pulling researchers back. um, so fantastic. Thank you so much. Thank you everyone for sticking around. We have enabled chat section here. You can type in your questions in the chat. Alternatively, you can also click this raise your hand button, and you can directly speak with we'll enable your mic once you click that, and then you can ask your questions directly to Dr. Iwahara as well. Okay, I see someone raise their hand. Let me enable their Marcel. Do you see? Okay, I'm gonna unmute Philip first. Marcel, can you unmute Philip? Philip, we can hear you, I think. Philip? Oh, can you unmute Philip? You can unmute yourself. Uh, it's okay. While you figure that out, let me go to Dr. Herman. Dr. Herman? You need to unmute.
This is Philip again. I've unmuted myself.
Oh yeah, perfect. Yeah, thank you. Yeah, you guys. Yeah, you have to unmute. Yeah, go ahead, please. Ask a question.
All right, that's a really interesting method, and thank you for publishing it. And I do have a few questions. Your reaction time is 90 minutes.
Right.
If I ran it for 2 hours or 3 hours, would I get more DNA, or is that just the lifespan of the enzyme, or what's
Okay, that's
that?
a great question, and we actually worked on that. So let me show something here. Okay, so do you see my slide for anyone?
Yes.
Okay, so here, this is basically sort of time course. Basically for this, that's the reaction at 1 hour, 2 hours, and 3 hours, and you don't see much difference, right? Because basically after 90 minutes, it basically almost completely consumes all the DNTPs. And in fact, if you add more DNTPs after such consumption, complete consumption of DNTPs, you get more products like this. And in this way, you can save materials, I mean, of the enzymes. But you can't, unfortunately, use high concentration DNTPs from the beginning. If you do it, some unusual process happens and you get non-specific amplification. Actually, we described such effect in our supplementary materials of the JAX paper. So you can find such information. So we have to use 0.2 millimolar DNTPs, but once DNTPs are all used up, you can add more to produce more materials.
Okay.
Really a great question. Thank you.
No problem. I got another question, if that's all right.
Please.
Go ahead.
Just one quick question that I wasn't going to ask. Is your 0.4 millimolar sample in a Shigemi tube, or is it a regular 500 to 600 microliter sample?
Yeah, we typically use just regular tube.
Okay. Another question then, if that's all right. I used to make use T7 RNA polymerase to make RNA, and we would often optimize in that case, it was the ribo NTP concentrations based on the composition of the of the RNA being produced. Have you tried, like, you're recommending the same concentration of each of the four?
That's another
if it was optimized.
Yeah, actually, you can change the DNTP concentration, and in fact, for telomere DNA, there is no C here. Okay, so this is a telomere DNA, but we still used DCTP, and that is just waste of the materials. And probably by using optimized ratio of DNTPs, yeah, we can get better yield.
Okay. Another question, if that's okay, coming from using T7 RNA polymerase, we would for each reaction we used to optimize the template concentration and the NTP concentrations. Now, we already talked about NTP concentrations. Is the template concentration very sensitive to production?
No, but actually, we found that 1 micromolar is almost optimal. If you use too much, somehow it didn't work very well, if I remember correctly. Anyway, 1 micromolar is close to the optimal, probably for almost any case. But I don't know. We checked only several different DNAs for that. But as long as we know, as far as we know, 1 micromolar is probably around the optimal range.
All right. Thank you. This is fantastic.
Thank you.
So, thank you, Philip. A couple more questions, Dr. Iwahara. Dr. Human, can you click on the mic? Then it will enable your mic. I have enabled from my side. You have to still click on the mic symbol so that you can your microphone will get active. Meanwhile, I have a question, Dr. Iwahara, from our chat. So what is the maximum length of labeled DNA we can produce with PaNDA without running into issues such as impurities and things like that?
Okay, so that's a great question. And in the JAX paper, we discussed that size limitation. And as far as we tried, 50 nucleotide is probably possible without changing the buffer conditions. But if necessary, we first would try lower salt concentration, because this salt concentration was optimized to basically suppress a 3' overhang. And if 3' overhang doesn't matter for you, you can use lower salt concentration, and that will increase the yield and also byproduct of shorter DNA. So the salt concentration is one thing we could optimize to extend the size limitation. But I don't think the current method, current conditions work for something like 200 nucleotides. It's just too long for this one. And the polymerase, this strand displacing DNA polymerase is not like Taq polymerase, and the dissociation rate is much faster, and because of that, the size is relatively short. But maybe by optimizing conditions, we can extend the size limit. Yeah, we are working on it actually.
Okay. Perfect. Thank you. Dr. Human, now you're active.
Yeah.
You can answer your question.
Junji, thank you very much for this great presentation. Really enjoyed it. And I think, to my mind, it's the best isothermal amplification method for DNA, so far as I know. And I'm very glad that we had you in this webinar, and to learn more about this kind of methodology. Actually, I have some question to the DNA polymerase, a very simple question. Have all DNA polymerases a strand displacement activity, or is it a special enzyme what you're using?
So this is just a special DNA polymerase, and basically you have to use, sorry, not here. You have to use DNA polymerase that has this activity. And BSD 2.0 DNA polymerase is one of the commercially available strand displacing DNA polymerase. But there are some other possibilities. But anyway, for example, if you use just typical PCR enzymes like Taq polymerase, this doesn't work because it doesn't have strand displacing activity. And optimal temperature range is different. Here there are two enzymes, right? Polymerase and a nicking enzyme. And a nicking enzyme's optimal temperature is around 60 degrees or so. So if you use, for example, this methodology together with Taq polymerase at much higher temperature, it doesn't simply because nicking enzyme doesn't work at such a high temperature. So 55 degrees is a good compromise.
Okay. Okay. Can I have a second question?
Sure.
Ravi? Yep. Okay. So my question concerns the fidelity of the system of the polymerases. If I understood properly, at the binding site or as a starting point, you have two enzymes which compete more or less for the same site. Does it interfere somehow? That's the one question. The second question is, due to the displacement activity, does that affect the fidelity of this enzyme, or are there numbers available about the fidelity?
Okay, so for fidelity, as far as we see in the NMR spectrum, we don't have any practical issues. If, for example, this is really error-prone, we expect to see multiple species in the NMR spectra, right? But we didn't see it. So probably the fidelity is practically at least acceptable. I don't know precisely the error rates for this enzyme. Maybe we can find it in the literature because ssDNA polymerase is well-studied polymerase. Regarding the specificity of nicking enzyme, that's an issue. Okay, so there is a basically so-called star activities of this enzyme, and the recognition is not really perfectly specific. In other words, it may cleave DNA at some different sites, of course, at a lower rate, but if you use, for example, very long DNA containing many different kinds of sequences, some part may happen to have a sequence similar to the recognition sequence of the nicking enzyme. So in that case, basically you see shorter products. But usually, even if you see shorter products, we can chromatographically isolate the desired product because shorter product comes out earlier in the chromatography. But anyway, the specificity of nicking enzyme is something we could address, or some companies like NAV can address. So if high fidelity nicking enzyme becomes available, that would be useful for us. And another thing is we tried some other nicking enzymes as well, but the enzyme called the enzyme called the enzyme called NT-BST-NBI nicking enzyme is the best. We tried some other approaches using different nicking strategy, but this enzyme works the best. And the reason could be that maybe competition between the polymerase and this enzyme occurs to a lesser degree. I don't know, that's just speculation, but depending on the enzymes, the efficiency of the amplification is quite different.
Yeah, thank you very much. It was a great presentation.
Thank you.
Thank you. Thank you, Dr. Human. Thank you, Dr. Iwahara. Do you have time for one more question, Dr. Iwahara?
Sure.
Okay, sounds good. So one of the question was, is there any issues you see PaNDA working with the modified NTPs as well, or should be pretty straightforward?
We don't have a good answer to that question because we tried just regular DNTPs.
Yeah.
But it might be possible to incorporate some unusual nucleotides depending on the fidelity of the enzyme. We just don't have any good information regarding that. I'm sorry.
Okay, yeah, that's good. Yeah, thank you. Thank you. Yep. So once again, thank you so much, Dr. Iwahara. We truly appreciate it. From Silantes side, I'm sure from our audience side as well. And we congratulate you on this exemplary work, and we look forward to many more publications like this in the future. Thank you so much.
Thank you.
Okay, thank you guys. Thanks everyone.
Thank you.
Bye-bye.
Bye.